All right, this activity that we're going to be doing that has to do with the size of a cell and its ability to transport material. All right, so we're not actually doing any like lab activity stuff. This is all going to be kind of measurements that have been done for you that we'll be using instead. All right, you'll have the Chromebooks today, and you should easily get this done in class today. The idea here is that this is not something that is going to require homework from you. All right, but that means, of course, you're going to have to be focused in class in order to get it done. Okay, we went over all this stuff yesterday on active transport, diffusion, and osmosis, so we won't spend much time on that. We'll just get straight into the lab activity itself. Okay, so give you the handouts here. Yep. Okay, so just quick review here. The main thing we're going to be looking at today, because we're talking about transport within the cell, is diffusion. Okay, because that's how a cell transports material from one place to another within the cell. Okay, because osmosis and active transport have to do with across the membrane, so we're not dealing with that today. Okay, remembering that it's passive, goes from high to low. Okay, that it is a bit slow over large distances. That's what we're going to try and investigate today. Okay, and that it is not targeted. All right. So the big thing we need to know here is that cells absorb nutrients and excrete wastes through their membrane, that is, their surface area. Okay? But they must transport everything they get through their cytoplasm, which is their internal volume. As a cell's size increases, the volume increases faster than the surface area, and transport times take much longer. Okay? Also, now comparably smaller surface area must supply and clean a greater volume. Right? Diffusion is too slow to supply or meet the needs of very large cells. As a result, cells do not reach large sizes. And that's kind of what we talked about yesterday. That's just a quick review of that. Okay, this was another version of that diagram I showed you yesterday with the little sectors coming from the lysosome to the mitochondria. All right, so the activity we're doing, okay, the problem we're investigating, how does the surface area relate to the volume in cells of different sizes? Okay, does dividing a large cell into smaller cells affect the surface area to volume ratio? All right, so our hypothesis here, okay, we want to generate a hypothesis about the relationship between surface area and volume in cells and how that can affect the functioning of the cell. So you might want to make a little note here, okay? In the if part of this hypothesis, okay, we should probably talk about diffusion being the primary method of cellular transport. Okay? In the and part of our hypothesis, we should talk about some of the characteristics of diffusion. Okay? It's passive. It's okay over small distances, slow over large distances, things like that. Okay, those should be part of the things that are in the and. Okay, in the then part, then we should probably say something about what will happen as a cell gets bigger. Okay, is its volume going to get bigger? Okay, is its surface area going to get bigger? What effect is that going to have? All right, that's how that hypothesis should go. You'll notice there's no design to this. It's because we're not actually doing a lab. We're not manipulating anything. Okay? There's no responding variable. There's no control variables because it's not a real lab. Right? So you don't have that part. Okay? So it's a little less work. That's good. Okay? The procedure goes like this. We're comparing these three imaginary organisms. Okay? One of them is a small, single-celled organism called micro. Okay? One of them is a large, single-celled organism called macro. And one of them is multicellular. All right? It's quite large, multicellular. We call it multi-micro because, well, it's made up of a bunch of little micro squares. Okay? Everyone follow me so far? All right. Now, just looking at these three cubes, okay, which one of them is going to function best? Micro. Okay? Micro is going to function best because it's got a very small volume, fairly large surface area. Okay? Diffusion will work well in something small. Okay? It's probably not going to work so well in macro because macro is very big and is essentially otherwise just like micro. Okay? What about multi-micro? Okay, well, there might be some competition for sure, but in terms of volume and surface area, is multi-micro going to be better than macro or worse than macro? Better, yeah. Okay, because it isn't one single cell, it's many. It's 1,000 actually. Okay, there's 1,000 little cubes in here. All right, it's not hollow, right? We're not just looking at the outside. The whole volume of this thing is filled up with tiny cells. And each one of those tiny cells is exactly like this one. Agreed? Okay, so that means that each individual cell should work okay. Everyone follow? Okay, that's the advantage of becoming multicellular. You can get big without having to deal with the problems of surface area and volume and diffusion being really slow because all your individual parts are small. Okay. All right. So we're going to have a 3-column table that's going to look something like this, and I think it's already inserted into the Google Doc that you're going to get. Okay. So we're going to have these cubes here. All right. Micro, macro, and multi-micro. We're going to calculate their surface areas, volumes, and surface area to volume ratios. You know what? All right, so while you're getting signed in there, okay, this is what your Google Doc's going to look like. All right, you'll have the spot where you'll write your hypothesis, okay, and then you'll have this chart. This is the chart we're going to fill in here, okay. So if you're to this point already, you'll probably want to put micro, macro, and multi-micro in for the cube names. Okay, so it's something like this. Okay. All right, now the dimensions of these cubes are that micro is 1 by 1 by 1, okay? And we'll say that that's micrometers, okay? Just so we have units, otherwise numbers are meaningless, okay? So 1 micrometer by 1 micrometer by 1 micrometer. Macro is 10 micrometers by 10 micrometers by 10 micrometers, and so is multi-micro, okay? The difference obviously is that multi-micro is made up of many small cubes, okay? All right, so if I want to calculate the surface area of the little cube of micro, how do I calculate the surface area of a cube? Okay, yeah, you're absolutely right on that. Let's say it the easier way. Length times width times the number of sides. Okay, that'll work out to the same thing, okay? But I think that might just be a little easier because it took me a minute even to figure out that, yeah, what you had said was right. Okay, so if I go length times width, the length is 1, the width is 1, and how many sides does a cube have? 6. Okay, so the surface area of this is 6 micrometers square, okay? Because it's a cube, okay? We're talking surface area, so our units become squared. Okay, so micro is going to have a surface area of 6 square micrometers. To calculate volume, I go length times width times height. What's the volume of the little cube? Oh, I really thought we'd get that one right away. 1 times 1 times 1 is 1. Okay, all right, so it's 1 cubic micrometer. So, is this a good situation? Lots of surface area and very little volume. Yes, that's very good, okay? Because diffusion doesn't have to go very far, and there's lots of membrane to acquire nutrients and get rid of waste and all that kind of stuff. That is a great situation to be in. Surface area to volume ratio is 6 to 1. All right, looking at macro, I got to do the same thing. For surface area, I've got 10 by 10, which is 100, okay, times the number of sides, 6. The surface area is 600 micrometers square. Okay, and volume is length times width times height. 10 times 10 is 100, times 10 is 1,000. Okay. Is this a good situation? Sorry, you're right, it is cubed. Okay, Is this a good situation? No. Okay? I have comparably less surface area and a very large volume. In fact, my surface area to volume ratio is now 0.6 to 1. All right, multi-micro is the one that's a little bit tricky. Okay, if I just go 10 times 10 times the number of sides, I'm going to get that the surface area for multi-micro is the same as it is for macro, and it isn't. Because multi-micro is made up of many little micros, all of which have 6 sides. What's the matter with that? Okay, so if it's made up of many little micros, okay, and I know the surface area of a single micro, would multi-micro have, let's say, let's look at how many micros are in that multi-micro. Okay, there's 10 on each side, right? So there's 1,000 micros in here, agreed? Okay, if each micro has a surface area of 6 micrometers square, then the surface area of multi-micro is 6,000. Okay, now when I'm calculating volume, I still do that. Length times width times height. What's the problem with that one? What? I unplugged them. Don't press the power button when you open the lid. Okay. Okay, so the volume of this one is the same as this one is, because they're the same size. Okay, so the volume of multi-micro is 1,000 micrometers cubed. Is this one have a good situation? It does. Okay, is its surface area to volume ratio the same as the little one? And it should be. It's made up of the little ones. Okay, so its surface area to volume ratio goes back to 6 to 1. Okay, so right there, the math is telling us being multicellular is good, because you don't have to worry about diffusion being too slow to transport stuff from one place to another, because you're made up of lots of little cells. All right, that was actually 5 marks of your lab report. You're welcome. Okay. What? Okay, just give it a minute. It's your own computer. There's not going to be much I can do for you. Okay? All right. For the analysis and interpretation, okay, you're probably going to want to write a couple of things down for this just so that it makes sense, because the wording of these is not great. Okay? For number 1, it's asking when the cube dimensions increase, and it says here from 1 centimeter to 10. We used micrometers, so you can just ignore that. Okay? So essentially, when I go from micro to macro, the size increase is 10. Okay? Or the sides increase by a factor of 10. How many times bigger did the surface area get? How many times bigger did the volume get? So basically, you just go back to your chart and you go, okay, how many times bigger is 600 than 6? And how many times bigger is 1,000 than 1? Okay, so I think you guys can figure that out. Okay. For number 2, okay, it's asking you to compare the 2 big cubes. Okay, so macro and multi-micro. They're the same size. They have the same volume. Okay, but it says how many times bigger is the surface area of multi-micro than of macro. Okay, that's what it's asking. How many times more surface area does multi-micro have than macro does? Okay, for number 3, it's asking which 2 of the 3 cubes, okay, if I make them bigger, will their surface area to volume ratio change? Okay, so if I make micro bigger, it essentially becomes macro. Okay, if I make macro bigger, it's just a way bigger macro. And if I make multi-micro bigger, it's obviously bigger, but it's going to fill up all that space with what? with more of the little micros that are all still the same size. That's just something we kind of have to know as background knowledge in order to answer that question. Okay? Number 4 is asking you to use this diagram right here, which is why I actually printed a sheet for you, because you're going to have to measure these shapes. Okay, so you'll need a ruler. I have some up here if you need to borrow one when you get to that question. Okay, so you're going to measure them and you're going to calculate their surface areas and volumes. Now, something you need to be aware of. They're not cubes. Okay, well, this one is, A is, but B and C are obviously not cubes. So you'll have to calculate, let's say, okay, the surface area of this side. And since the bottom is the same, multiply it by 2. Okay, the surface area of this side, and the other side is the same, so multiply it by 2. Surface area of this side, multiply it by 2, and add them all together. That will give you the total surface area of that shape. Okay, and then the volume is still just length times width times height. Okay, and you'll have to do the same thing for C. All right, you can't just go length times width times the number of sides, because the sides aren't all the same size. All right, everyone follow me on that? Then you're going to calculate the surface area to volume ratios of those 3 things to see whether shape affects the surface area to volume ratio. All right, because I'll tell you right now, the volume of those 3 shapes is very close. Okay, they all have a very similar internal volume. All right, but what we want to find out is if you change your shape, can you change your surface area? Okay, and as a result, make diffusion actually still work. Okay. Okay, for number 5, it's asking about these 3 example cells. So a liver cell, which is kind of like cube A. Okay, a skin cell, which is kind of like shape B here. Okay, and a nerve cell, which is kind of like C. Okay, and it's asking you how have those cells adapted, okay, to deal with the problems of surface area to volume ratio. So I mean, your liver cells obviously are in your liver. Okay, they're well supplied with blood. Okay, your liver actually has probably more blood in it than any other organ in your body. Okay, so there's lots of nutrients going in there all the time. Your skin cells, obviously, they only get supplied with nutrients on how many sides? Yeah, 1, 2, maybe 3, okay, but obviously they're not getting anything from the outside, so, okay, they have to have a certain kind of shape. And nerve cells can be up to 1 meter long. Okay, you have some nerve cells that essentially run from the base of your brain to the base of your back, okay, down your spinal cord. All right, they can be up to 1 meter long, but they are probably very, very thin. Right, and not probably, they are very, very thin. Okay. Kind of like C, right? So you kind of got to put those 2 questions together a little bit. Okay, and then for number 6, it's asking about the red blood cell here. Okay, and the red blood cell you can see has an indentation in the middle. All right. Now, what is the job of your red blood cells? What do they have to do? They have to deliver oxygen. They have to carry oxygen, and where they carry it is on their membrane. Okay, that's where the hemoglobin is. Okay, so the hemoglobin is all over the surface of the cell membrane. So anything that the red blood cell could do to increase its surface area would probably make it more effective. Agreed? Right? What you have to look at is, does that shape do that? Okay. And then for number 7, okay, this is kind of the big, most important question in the analysis, and it's what we've been talking about the last 2 days. Most materials get from place to place in the cell by diffusion. What effect does a low surface area to volume ratio have on intracellular transport? Okay, so you kind of, that one should be fairly easy. All right, then your conclusion, and that's it. You don't have any evaluation because there's no sources of error here. We didn't take any measurements. All right, questions on that? All right, you have got 50 minutes. Okay, to get that finished. I don't think you'll have any trouble with that since we've kind of done all the calculating already. You just have to do a hypothesis, answer analysis, and do a conclusion. Yes, on the chart.